Assalamualaikum warahmatullahi wabarakatuh. My name is Amir Ashraf bin Ahmad Azam. Today I want to explain the experiment of bacteria enumeration. So first, I will show how to observe the experiment from viable plate count. So first, click on the dilution tube right of test tubes to bring them to the table. And then click on the water samples to bring the samples to the table. Now click on the eyedroppers to withdraw 1 ml of sample 1 fountain water and dispense this to the first dilution tube. And then click on next when this initial transfer is finished. Click again on the eyedropper to withdraw 1 ml from the first dilution tube and dispense into the second dilution tube, and subsequently withdraw 1 ml from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is complete. The dilutions for each of the 6 dilution tubes can be summarized in the image below. Dilution tube 1 has a 1/10 dilution with the dilution factor of 10. The dilution factor for each of the tube is listed below. Next, we will be inoculating agar plates with the last 3 broth culture dilutions. Click on the agar plates on the shelf to bring them to the table. Now, click on the eyedroppers to transfer 1 ml of dilution 4 to plate 1, 1 ml of dilution 5 to plate 2, and 1 ml of dilution 6 to plate 3. Next, click on the pencil to label agar plate 1 with a dilution factor 10,000, plate 2 with a dilution factor 100,000, and plate 3 with a dilution factor 1 million. Click on the agar plates to place them in the incubator at 37 degrees Celsius for 48 hours. We will now need to perform the same dilution and inoculation steps for each of the test samples. The process is the same for each sample, and we will assume the process of dilution and inoculation has been completed for all 4 of the water samples, and then 48 hours of incubation time has now been completed. Next, click on the incubator to bring all of the inoculated agar plates to the table. Each of the groups of inoculated plates is labeled with the source of their respective samples. A key for the sample as is given below. There are 4 samples, which is fountain water, white fountain water, river water, and boiled river water. We are viewing the agar plates that were inoculated with fountain water. The dilution factor for this plate is 10,000. For this plate is 100,000. and for this plate is 1 million. Next is boiled fountain water sample. I think same is like as fountain water sample, which is 10,000, and this is 100,000, and this is 1 million. Same for river water. And also boiled river water. That's all for variable plate count. So next, I will show the experiment of direct count. So for this experiment, first click on the water samples to bring the samples to the table. And then click on the methylene blue bottle to bring the dye to the table. Now click on the top of the methylene blue dye to withdraw 1 ml of the dye and dispense this to 1 ml of each of the water samples. Next, click on any one of the numbered water samples to add 1 ml of the sample to the Petrov-Hauser counting chamber for viewing and counting using the microscope under 400 times. We will need to view all 4 of the water samples, which is fountain water, boiled fountain water, river water, and boiled river water. So first, we could see for the fountain water, click on the microscope to bring it to the table. Next, click on the slides to bring one of them to the microscope. And then click the eyedroppers to transfer 1 ml of water sample 1 to the slide. We are viewing bacteria from sample 1, fountain water. So this is the results. Next is boiled fountain water. Do the same procedure, same like we do on the fountain water. So this is result. Next is river water. So this is result. So the last one is boiled river water. So this is result for boiled river water. So that's all for direct count experiment. So for the last experiment that I will show is durability count. So for this experiment, first click on the water samples to bring the rack of samples to the table. There are only two samples of water we will test. Sample A is faucet or fountain water and sample B is river water. Next, click on the sterile dilution tubes to bring them to the table. So next, click on sample A to move the sample to the dilution rack. Next, we will perform the dilution by transferring 5 ml of the original sample to the first dilution tube labeled as 1/2. Then we will transfer 5 ml of the 1/2 dilution to the tube labeled as 1/4 and so on until the last tube labeled as 1/16 has 5 ml added to it. Next, click on the blue eyedroppers to perform the dilution. There is a tube labeled as blank which contains only pure sterile broth with no bacteria. Next, click on the spectrophotometer to bring the machine to the table. Next, click on the mode button to set the machine to absorbance mode. And then click on the dial to set the wavelength to 520 nanometer. Next, click on the blank to insert it into the spectrophotometer. Next, click on read to view the absorbance for the blank. The blank will read 0 as there are no bacteria in the solution and thus no absorbance of light. So next, click on one of the dilutions in the dilutions rack. Once the dilution has been inserted into the spectrophotometer, click on read to view absorbance for that dilution. Record your value for the dilution that you have selected in data table 3 for fountain water. One at a time. Click on each of the other dilutions and then click on read to view each of the absorbance. So this is the result. So next is sample B. Click on sample B to move the sample to the dilution rack. Next, we will perform the dilution by transferring 5 ml of the original sample to the first dilution tube labeled as 1/2. Then we will transfer 5 ml of the 1/2 dilution to the tube labeled as 1/4, and so on until the last tube labeled as 1/16 has 5 ml added to it. Next, click on the blue eyedroppers to perform the dilution. Click on the spectrophotometer to bring the machine to the table. Click on the mode button to set the machine to absorbance mode. And then click on the dial to set the wavelength to 520 nanometer. Click on the blank to insert it into the spectrophotometer. Next, click on read to view the absorbance for the blank. The blank will read 0 as there are no bacteria in the solution and thus no absorbance of light. Do the same procedure as before as the sample of A. So this is the result for sample B. So that's all for turbidity count.